Now that you learned about transcription, it's time to learn about translation, the second phase of protein synthesis. The first thing that you need to know is that translation, unlike transcription, occurs in the cytoplasm, on the ribosome, or on the endoplasmic reticulum. Think of the ribosome as a hamburger bun. There's a small lower subunit and a large upper subunit. The mRNA strand travels between the two subunits for processing. There are 3 sites on the upper large subunit: the A site, the P site, and the E site. The A site is like a waiting room. The next codon that travels through the ribosome waits there to be decoded. It moves on to the P site, where transfer RNA reads the codon, and then the transfer RNA moves on to the E site, where it eventually exits the ribosome without its amino acid. The steps in translation are as follows. First, like I mentioned, mRNA slides through the small and large subunits of the ribosome. The mRNA transcript is read 3 bases at a time. We call that a codon, a sequence of 3 nucleotides. Translation starts when the codon is at the P site. The start codon, the sequence on the mRNA to start translation, is always AUG. The transfer RNAs bind to amino acids in the cytoplasm according to their anticodon sequence. The transfer RNA then binds to a specific codon on the mRNA. It's complementary to the mRNA, and add their amino acid to a developing protein. Here is a picture that depicts all the steps. Here are amino acids. Amino acids are being picked up by transfer RNA that has a specificity for that amino acid. The transfer RNA then comes over to the ribosome and hangs out in the A area and meets up with the mRNA. Notice that the anticodon is complementary to the codon on the mRNA strand. The mRNA bound to transfer RNA moves to the P site, right, and releases its amino acid and creates a chain that will become our protein. The tRNA moves on to the E site, leaves the ribosome, and is back out in the cytoplasm to pick up some more amino acid and start the whole process over again. So a little bit about the genetic code, right? Each codon codes for a specific amino acid. There are 64 combinations of the bases, but only 24 amino acids. So some codons code for the same amino acids. This slide shows 2 different schemes for figuring out the amino acid that's coded for within an mRNA codon. So for instance, if our codon on the mRNA is G, C, or A, G, C, and A, what we do is start at the center. Here's G. In that region, right, the second quadrant, let's say, if we're thinking about geometry, the next letter being C, we'd move to here. And then if A is the third letter, right, it falls somewhere in this region. That means that the amino acid that mRNA sequence GCA stands for is ALA, or which stands for alanine. If we use this table form and we look up GCA, our first letter here is G. Our second letter, so we go to the column for C, we're in this region, and our third letter is A, right? Then the second one from the bottom, right, it's alanine. We'll be doing this in the activity that we do later this week. So ultimately, translation forms proteins. Proteins, again, are amino acids that are bonded together The transfer RNA reads codon in the mRNA and links the corresponding amino acids together to make a polypeptide chain. Eventually, the messenger RNA will reach a point where a stop codon reaches site A, and then translation stops. The new protein, the long string of amino acids, breaks off, and translation is over.
Okay, so how does translation work? Let's kind of orient ourselves because this diagram seems crazy. Here's our messenger RNA. So this is going to be our messenger RNA right here. And so it's moving through a ribosome. So a ribosome is where we make proteins. It's going to have two parts to it. It's going to have a small subunit, that's this bottom part. It's going to have a large subunit on the top, and then the messenger RNA is going to flow right through it. Every 3 letters in the messenger RNA is going to code for 1 amino acid. And the reason why is since we have 4 letters in DNA, 4 letters in RNA, A, T, C, and G, or A, U, C, and G if we're talking about messenger RNA. Every 3 letters are going to code for 1 specific amino acid. And the reason it's every 3 letters is it gives us enough variety that we can cover all the amino acids. So speaking of amino acids, where are they? The amino acids are going to be these little balls, and they're on top of another type of RNA that's called transfer RNA. Transfer RNA's job is to transfer amino acids from the cytoplasm to the ribosome so we can attach them on to this growing polypeptide chain, which is going to become this protein. And so basically, what does a transfer RNA do? Let's just kind of look at one. It's going to grab its amino acid right here, and then when it's its turn to go to the ribosome, it's going to bond to the ribosome like this, and then it's going to drop off its amino acid. And so it's basically dropping off this amino acid. It's then going to flow out into the cytoplasm to grab the next amino acid. Now each tRNA is going to grab a specific amino acid, and it's going to have 3 letters on the bottom of it. We call that the anticodon that's going to bond to the codon that's found in the messenger RNA. And so basically this occurs over and over and over and over again until it gets to a stop sequence on the end, and then we're done with that protein. And so I'm going to start this animation over here. And so basically, let me kind of talk you through it. This down here is going to be the small subunit. This on top is going to be the large subunit. And then we can see that we have all of these, they're blue, these dark blue, are coming in, and they're actually bringing their amino acids with them. Now there's an enzyme that attaches that on, but basically these dark blue tRNAs are each dropping off an amino acid, and then they're taking off into the cytoplasm to pick up another amino acid. You can see right here that the messenger RNA that came from the nucleus is just feeding through the middle of that ribosome. And it's a site where we can actually have the tRNA come in. Now if we watch this animation here, this is a ribosome that's bound to the endoplasmic reticulum, and so it's going to attach to the endoplasmic reticulum, and that growing protein that was growing inside that large subunit is actually going to grow into the middle of this endoplasmic reticulum, and so we can modify it or do things with it. And so basically this is translation. So translation, if we go back to our analogy again of the cooking analogy, basically the ribosome serves the purpose of the chef, and so it's taking together all these different ingredients, all these different amino acids, and putting it together to make a protein. So we're translating that message into a functional protein. You can see the protein coming out on this side. Okay, what I'm going to try to do here is actually decode a gene. So we've got gene found within this stretch of DNA. So this is a double helix, so we have complementary strands. And so basically what I'm going to do is the process of transcription. So I'm going to go from DNA to messenger RNA. And then I'm going to try to translate that message, and I'm going to go from messenger RNA, or through the process of translation, I'm going to go to the amino acids found in the protein. Okay, so basically I'm going to play the role in this first line of RNA polymerase. So in the cytoplasm, RNA polymerase is going to look what's at the DNA, and it's going to put a complementary RNA. So in the DNA, A goes to T, but when that unwinds, the T will actually go to A, in the messenger RNA. T goes to A goes to U. Remember, we only have uracil in messenger RNA. G goes to C goes to G. And so if I just keep going, this right here would be an A and a C and a G, and this is going to be a G and an A and a G, and this is going to be a C and an A and an A, and then this is going to be a U and an A and an A. And so every 3 letters in this messenger RNA is going to be what we call a codon, because it codes for a specific amino acid. Now if we want to do the actual translation and figure out what amino acids are created, well, I can't do this from memory. I have to use one of these standard genetic code decoders. Some of them will be circular, some of them will be like this. But basically what you do is you look at the codon in the messenger RNA, so AUG, and then I just find that down here. So here's AUG right here, so that's going to be methionine. So methionine is going to be the first amino acid. Now methionine is special because it also starts all genes. All genes are going to start with methionine. Sometimes we trim that off later, but basically that's going to be our start sequence. Let's go to the next one, ACG. So I find ACG. This is threonine, so that's going to be like this. If we go to the next one, it's GAG. So GAG, that's glutamic acid, so that's going to be GLU. If we go to the next one, it's CAA. So let's find that. CAA, that's glutamine, so that's going to be GLN. And then our last one is going to be UAA. So when I find that, you can see it's right here, UAA. That's going to be a stop sequence. And what does that mean? Well, that's the end of the gene, so that's not going to actually code for an amino acid. It'll actually put in a release factor so the whole thing can break out. And so this is going to be our first amino acid, our second amino acid, our third amino acid, our fourth amino acid. This whole thing then is called a polypeptide. And again, it would fold into a specific protein to do a specific job. Now, most of them are going to be much longer than this, but what I've shown you is how DNA becomes messenger RNA. Amino acids will eventually form proteins, and now that whole thing is eventually going to make you, because you're made up of proteins. And so that's transcription, that's translation, and I hope that's helpful.